Okay, so the last segment is about predicting functional variation, right? So we went through mapping, we went through a relationship between generalized and phenotype, general properties of genetic variation. Note is not on. 1, 2, 3, 1, 2, 3, didn't work. So we went from general properties of genetic variation relationship to phenotype making genes. So now it's the very basic properties of function, what actually happens at the level of molecular function when you introduce variation. It's one lecture, there is a lot in the field, so what I decided to do, I decided to do 2 segments. One is to talk about missense mutations in proteins, so those variants that change amino acid at the protein level, and talk about non-coding regulatory variation. So I'm not going to talk about splicing. I wanted to. This morning I covered my slides, there's no chance. And I wanted to talk about estimation of the effect of loss of function variance, and I won't do that as well. So we can talk offline if any of you are interested. Okay. Moving to coding variance. We'll start, I'll mention experimental techniques. Primarily, I'll be focusing on the computational prediction because this is the topic of the school and this is what we're doing. And we started this, I believe, 20 years ago by now. It's hard to say. It was a long time ago. So we started working on this problem, I think, alongside maybe John Malt and Pauline Ng, looking at the possibility to predict whether the variant will significantly impact protein structure and function or it wouldn't. And the information we were using was evolutionary information coming from the sequence alignment and structural information and information from the annotation. So the evolutionary information, now what we think about evolutionary information. So there are a lot of methods, very new methods that heavily rely on evolution. This is actually working, structure works much less accurately, so structure doesn't work that well. So what's happening here? There is the assumption that if I have this site, right, and I have a variant in a human, and I have looked at other species, for example, here is dog and fish, I assume that this pattern of amino acid changes and the rate of amino acid changes. So the rate with which evolution happens at the site and the types of amino acids I see are stationary, right? So I assume that these other species are informative about the human condition. Generally, I'm making an assumption. In evolutionary terms, this is the assumption of constant thickness landscape, right? So what I assume is that if certain amino acid is fine for human protein, doesn't change function of the human protein, doesn't kill function of the human protein, right? It's the same for dog and the fish, and if something is changing function of the fish protein, it changes function of the human protein. Why is this assumption not fully correct? So one, fish lives in a different environment. For some proteins, environment matters. If you don't know, fish lives underwater. Fish never gives 5 lectures on their own, like never. This is not what fish do. Okay? So humans are different from fish, right? The other thing is that there is epistatic interactions, what we call dependencies, because fish has many other changes in the genome. And I'll show real fish experiments, you'll see how it looks. And these changes in the genome introduce new context for this position, right? So I cannot look at this position alone because I'm uninformed by all other changes along the sequence. Now, if I make an assumption that this doesn't matter, and we'll talk how much it matters actually in reality, then I can look at the site and I can build a probabilistic model. And usually I don't have enough sequences, so what I have to do, I have to model phylogeny. And because I don't have enough observations, I have to be Bayesian, so I have to introduce some idea of priors and all this usual Bayesian tricks. So that's the idea of using evolution. And I basically just said this. So you can read what I just said. Okay. The other source of information is protein function. And at some point, many years ago, I think John Moll's group published the observation that most disease mutations happening in proteins change protein stability rather than function. Right? And the reason for this is that mutation target for mutations changing stability is much greater. It's much easier to hit protein in a random place and make it unstable rather than change specific component of the function. So that sounds like great, but estimation of delta G, which is free energy of folding this stability, is very difficult, remains very difficult. So this whole problem is very hard. So far, everybody is enthusiastic about the structure. Helps. Again, not as fruitful as at least I had expected when we started working on this. Now you can go from sequence to structure, so that's almost a moot point, but I think being motivated by structural properties of the sequence will still help. Okay, this is now already 13 years back, so this is the one version of our own software looking at multiple sequence alignment, trying to think about distribution of amino acids and rate of evolution, looking at properties of amino acids and things like structural effects like crystallographic B factor, for example, estimating mobility of the amino acid. Related method, for example, SIFT is based purely on multiple sequence alignment. Roughly same idea, different algorithm. It's different way to treat phylogeny, different way to treat priors. And I think I appeared the first version almost the same time as ours. And then people started developing umbrella methods. So what the observation in the field is that you build these methods, you build classifiers, and they don't produce identical results. The observation is, even if they use the same data, to move and combine them into an umbrella method, right? So to take a method and feed many methods. So very popular method is CAD. And CAD is popular for one smart idea. They take all variants of all types and put them on the same scale. So they can predict missenses, but also nonsenses and non-coding, and it's very convenient to use. And the idea comes from the fact that what they do is they are one cluster. They have positive and non-labeled, in machine learning terms, structure. So they are saying, okay, we have variants we observe, and we have a proxies, variants we simulate and never observe. Now this is a very powerful idea. It also leads to a lot of issues, right? Because when you simulate, you're not capturing all the data. You're not capturing intricacies of mutation rate, intricacies of selection, bias gene conversion, whatever other biological effects happening at the variation level. That's a popular method. So newer methods, this works actually pretty good. It's called REVEL. And REVEL is an umbrella method using random forest. Random forest is extension of decision trees. And decision trees everybody likes because the concept is very simple and they capture dependencies, but individual tree doesn't work very well. So what you can do, you can create multiple trees, train them on the subset of data and let them vote. And that's what random forest is, and this is a reasonably popular method as well. I would mention this 4. I would mention Verity, which is using gradient boosting, which is also a structure over decision trees, and specifically focusing on de novo mutations and ultra-rare variants. There are multiple other methods. These are mostly new. These are, I still call them, so first most of them are umbrella methods, so they feed a lot of methods into them. Second, I call them old school because they don't use large language models, so they are not like ChatGPT, they are more like classic statistical techniques. And this is what the community is using the most. I think the tide may be shifting, as I'll mention in a second. Okay, so what are the problems? Why can't you predict exactly? And I'd like to spend a little bit of time here because I discussed with some of you some of the aspects yesterday. And I think this is important because I think a lot of method developers just don't understand what is the problem they're trying to solve. Sorry, now, it was not meant to be disrespectful about other developers, and I know you're recording, so I take this back. Okay, so one issue, which is a very serious issue, is presence of weakly deleterious variation. So what I mean by that? If I look at comparative genomics, or if I train my model, irrespective of comparative genomics, on proteins that I see in the database, I train on the variants that fix in the populations, that are canonical variants in the population. If I have very small effect, you have a mutation which is deleterious in evolutionary sense. It might change function very little. Natural selection is a very powerful force. So it cannot be a truly pathogenic variant or variant that really knows down function, so you would not guess a clinical presentation or experimental outcome well. And this is my old slide. I decided to include it because we discussed it with some of you. So this is very classic stationary population genetics diffusion theory out of God knows what, 1960s. And that's equation for fixation probability. So this is the chance that a new mutation will be fixed in the population. I would observe it as a fixed variant in everybody, as function of selection coefficient. So look what's happening. So this is relative to neutral. So this means that what is my selection coefficient? Selection coefficient is reduction in fitness. So 10 and minus 4 means that individuals with this variant have on average 9,999 progeny compared to individuals without mutation who would have 10,000. Right? So you lose 1/10,000 of fitness due to this mutation. It's not a monogenic disease. It's not a serious effect. Nobody really cares about this. However, evolution does. So what's happening is there is this change, which we call drift barrier in population genetics, at the point of reciprocal of effective population size, which is roughly 10,000 historically for humans. So this means that if your selection coefficient is above this value, you're never going to fix in the population, never ever. So it means that if I'm training the method on sequences I observe or on comparative genomics data, I don't have any resolution whether my mutation is lethal, my mutation is 10 minus 3, effect on fitness of 10 minus 4. It's all indistinguishable. At the same time, if I'm below 10 minus 5, I'm neutral. Selection is not effective. I would see those variants all the time. Right? I just don't know. So the range where the whole game is very narrow. So the problem is that without population data, without estimating the effect from variants that segregate in the population rather than from sequences that fix or exist, we lose this dynamic range. We cannot say anything. So that's problem number one, and that's an important problem to solve, and I don't think we have much progress, so we're trying to move in this direction. I know some other groups are trying to move in this direction. So the other problem is I told you about dependencies between sites, or what we call in evolutionary genetics epistasis. This is from our paper with Alex and Todorkovsky from like 2002 or something like that. This is just an old story, but I keep the slide for the following reason. Again, I don't know whether inference is right here. I'll show you experimental data where I'm sure about the inference. Here I'm not. But this is what's happening. This is human beta hemoglobin, and at least the database says that mutating beta 20 for glutamic acid is a disease mutation, is a deleterious mutation that kills hemoglobin function. Now my problem is that if I look at comparative genomics, I go to the horse protein, and glutamic acid is here in the horse protein. Now what happened? Do I think that the horse has unfunctional hemoglobin? I mean, I look how a horse can run, and like how I can run. I mean, a horse has everything's fine with a horse. Now, probably what's happening is that the second site, right? So glycine 69. This interaction is nice and good. In the horse version, it's histidine. It's probably protonated. We have a nice salt bridge potentially forming here. Okay, so there is. You can only put glutamic acid on the horse background. You cannot put glutamic acid on the human background. Okay, so that's an issue. And if you look at the single site evolution, if you look at comparative genomics, there is no way for you to tell. So what we did, and this is still a few years back, so this is a quick story. It's a paper led by former grad students in the lab, Daniel Jordan, and in collaboration with lab of Nika Katsanis. Was published in Nature, I think 2015 or 2016, something like that. So we looked at a whole bunch of mammals and general vertebrates. Oh, That's again moving to a different computer. Anyway, so this is the observation. So for mutations that are annotated as pathogenic in human, potentially killing function of the human protein, about 5%, 5-6% are detected in mammals. Now some reviewers told us, oh, how are you sure about annotation, alignment, stuff like that, so we read a whole bunch of literature. It's probably some of them may not be exactly correct, but majority are very valid. I mean, this is a very solid observation. So what we postulated that a lot of this is happening because of a different substitution in the protein of the species. Right, so it's either happening in the human lineage or is happening in the protective mutation happens in the lineage leading to this mammal. So Daniel wrote a simple statistical model, and we believe that within mammalian tree that's usually a single substitution. I didn't believe in that. I believe in the model that there are multiple, right? So like multiple effects that overstabilize the protein. Doesn't seem to be the case. It's a one compensation. If it's one compensation, we can find it, but we have to go and look at every single change that we predict and see if it works. And we decided to work with Nico because they had a zebrafish model and they can inject the embryo. And what's important about zebrafish model? We frequently know it's in cis because look what's happening. So we have, maybe I'll show this. I wouldn't go through. Oh yeah, I can probably go through this slide. This is a model for one disease. It's Barrett-Biddle syndrome. There are many models in zebrafish, so the major story will be in a different model. But this is how the test works. So they look. This is the zebrafish embryo. I'm totally fascinated by the fish system. You never can guess what human phenotype how it's going to be modeled, right? So Barrett-Biddle syndrome. Again, I don't really know details on manipulation, but it's also developmental delay. So this embryo is developed normally, but if you have small or larger increase in the angle here, this embryo is underdeveloped. And they believe is this models human phenotype. So what you do, you look at the embryo and then you knock down the gene. So the fish gene, lack of the fish gene, gives you the phenotype. And then you take human mRNA and rescue it back. So you know human gene works on the fish background because you actually get the normal fish back by injecting human mRNA. And then the observation was the following, that this is the assay to test human mutation. You can put human mutated RNA, and it does not rescue. So this is how Nico's lab proves that these mutations are pathogenic. And you see this mutation actually reducing function of the fish, reducing function of the protein and giving fish phenotype. The issue is that this human mutation is sometimes native variant in the fish. Do you follow? Right. So what I'm doing is I remove fish gene. I see the phenotype. I give it human mRNA. Fish is fine. I give human mutant mRNA, and fish is not fine. But this mutant is the mutant existing on the native fish original gene. So this means that there is something else in the sequence of the fish gene that enable this mutation to be okay in the fish, on the fish background, and not okay in the human background, in the same fish system. So what we suggested is that we take for every gene, and we would take, so there's human gene with disease mutant, right? So that doesn't produce the rescue, and then we can have a double mutant, right? And if there is no suppression of mutation, it doesn't produce the rescue. But if double mutant produces suppression, then the second mutation completely compensates for the first. So that's the strain. So the idea again, I take a human gene, right? I knock down the gene, I see phenotype. I rescue phenotype with the human gene. I take human disease mutation, I see that it doesn't rescue. So human mutation is pathogenic because it doesn't rescue fish phenotype here. And I take the second mutation, I take human gene with two mutations, and this is a back functional protein. Okay, so we found a bunch of this. It's usually one or two changes. And interestingly enough, we would not be able to predict them structurally. These are very distant mutations. I have no idea how biochemistry works, but the effect is very clear. Now the interesting story is, again we're missing some information here. It's from Stefan Frangakis, who ran the experiment. So there was a patient in the clinic at that time in North Carolina, or where the lab was, and she presented with multiple phenotypes. It was a clear de novo mutation, and this gene was an obvious, Tyson, nobody looks like compound heterozygous in Tyson, that's the longest gene in the genome that's false positive for every single analysis. But this gene makes total sense, right? In this phenotype, it's a neuronal gene. And Nico's lab ran 3 different phenotyping assays, and they all worked very fine. So we know that this gene is a causative gene. The issue is that if you look at the alignment, her mutation is variant for methionine. So that's complete reversal to mammalian consensus. Every mammal but apes have methionine. So people didn't believe this variant, and what we show what Nico's lab showed experimentally, we take this methionine, right, and we take some of the other mutations happening in any of the species, right? I think there are 2 in total. They completely rescue the effect of this mutation. So variant is actually pathogenic on the human background, is not pathogenic on any other mammal background. This was published, we were pretty confident, but you still have this nagging feeling. And 2 years ago, there was an email that people found 2 new patients looking exactly like her with mutation in the exact same nucleotide. So we know this story is true. Okay. So what's happening is why it's difficult to predict computationally, especially from comparative genomics, the effect of mutation. 2 effects. One is I don't know about the scale of the effect because the observations don't tell me. The other one, there are correlations on the sequence that I cannot analyze. There are new methods. So we're working on the population level constraint measures. This is too early to present. And we hope that we can resolve the problem of distinguishing between strongly and deleterious mutations. And now there is booming of machine learning techniques. They try to solve the problem of interactions because this is what they are learning, right? So ideally, large language models learn interactions. Bunch of methods. So out of Debbie Marks and our Debbie Marks, Fraser et al. A year and a half ago, this method Eve, and that's based on variational autoencoder. For those of you who are computational machine learning aficionados, you probably love autoencoders. Everybody does. This method works is sort of okay. So it's it puts everything in this latent space and computes a model for the whole sequence, including every interaction. There are new developments, so this is very prad. I found it in the bioarchive. I'm not sure the paper's out. So what's happening now, we have protein structure prediction, right? So large language models are able to predict protein structures, and there are new methods. So one is coming from a company called Meta. Some of my family members are using Facebook, so I know what the company is. So they generated this method. So what the observation is quite interesting. Again, I'm sorry for the quality. I should have just saved this PDF. I think that was a mistake. I showed this problem. It's very nice in my laptop. So what's happening here, interestingly enough, is that looking at embeddings in language models. So you change embeddings for amino acid, and this works much better than running the full model and seeing the likelihood of structural change. I don't know why, but just in their results, looking at embeddings is working better. There are multiple efforts looking at AlphaFold, right? So this is again because the structure prediction is not a physical method, it's not related to stability, it's not related to any physical property, it's very hard to do. But there is some progress in the methods, and some people report reasonable results. Again, if you look at this accuracy, it's not close to 100%, right? So it's still way to go. I mentioned yesterday, so I'm somehow became an author of this paper. So this comes from Illumina. Again, we discussed cloud computations with some of you. All these methods come from large companies because there is no money in academia to run any of this, right? Well, maybe in Turkey, in American academia, there's no money. It's like to run this type of calculation. So this is done in collaboration, well, it's done by Carl Farr's group, and they somehow ended up on the project. So they first, because they are Illumina, they sequenced all private species, and this really helps. They use transformers and autoencoders within the system, and they use structural information and evolutionary information, so everything goes into the sync. They report reasonable increase in accuracy. For example, this is all of the CleanMar data sets, so these are non-pathogenic mutations. These are unbiased, these are de novo mutations in DDD, so these are de novo mutations in one of the consortium, much larger, more successful consortium than what I showed you. It's in the UK, it's about developmental disorders, so they show greater correlation compared to random for de novo mutations in autism and again, if you look at the amount of increase compared to standard methods, some of this are developed originally many years ago. It's not a stunning improvement, right? It's not changed in the game, but that's definitely an improvement, and this is where the field is moving. This is just out in Science together with other papers. The field's moving very fast because I cannot tell you about the papers I'm reviewing for those journals, but it's more and more of the stuff is coming out. Okay, so this is roughly the state of the art in predicting effect of missense mutations applications. One application is in the Mendelian genetics. We discussed that. I want to prioritize important variants in the analysis I presented at the last lecture. And we also discussed this rare variant association studies burden test. And this is what people do. Again, the idea is that if I can focus on functional variants and ignore benign variants, my association test power goes up. And that's quite clear. Even if I combine variants with vastly different effect sizes, if I combine real effect but weight them identically, this cuts down statistical power of the burden test, right? So that's probably relatively clear because I'm combining variants together to draw statistics. This is on the Mendelian data set, so this is developmental disorder, that's from private AI 3D. You see the increase in observed expected ratio for missense changes in de novo mutations in developmental disorders, and you see that if you run prediction compared to no prediction, right, so the effect goes up quite a bit. They probably missed something because this is benign missenses, and this synonymous provides exact control, right, so that's observed expected is exactly 1, and there is a little bit of inflation here. This is the effect of true nonsense mutations, right? So so obviously these guys are not as bad as true nonsenses. This is the UK Biobank. This comes from Regeneron on sequencing in the UK Biobank, and again they show the value of predicting running shared tests between loss of function and predicted missenses. They run pipeline with 4 traditional methods, and this increases the signal. So that's the major application of this in human genetics right now. Experimentally, again I mentioned yesterday deep mutational scanning. There is a lot of interest. It's a complicated proposition because you have to construct every mutant. You have to measure the functional effect of every mutant. You need an assay specifically for a protein. And that's roughly the whole idea. This specific one is for interaction, so you need a functional readout, right? If you have a complex, you have a functional readout. If you don't have a complex, you don't have a functional readout. And you can measure this. So that's interesting because what these methods can do, what computational methods can't. Because experimental methods are also noisy, right? So we're probably in the state of the world where computational methods are competitive with respect to experimental methods. However, in this paper, this is in Cell, I think it's this year, maybe it's last, I think it's not a mistake, it's for this year. So MC4R, it's a melanocortin receptor, so that's a classic gene for childhood obesity, right? So people don't feel that they full, they can eat infinite amount of food and never get full, right? So that's the issue with this receptor. Now what they did is they looked at 2 functional readouts, and there are 2 observations here. So one thing computational methods don't do well, they find gain-of-function mutations. A bunch of these are gain-of-functions. They are shown in this picture. When you look at this with your biochemical intuition, I have zero idea how to predict it. Right? So a lot of these gain-of-function variants are within transmembrane helices. I'm flying blind here. We try. I just don't know. Another thing is that they measure two assays based on two functional readouts from the protein, and they don't produce identical result, and one is important for the phenotype, the other one is not. So what I know from this assay, I told you computationally, because of the population genetics phenomenon, because of fixation and driven barriers, I don't know the scale of the effect. Here I do, because I measure it experimentally. I know direction of the effect, very difficult to do computationally. And I can measure different components, different types of function, because when I do computation, I literally measure force of evolution, right? That's what's happening. By the way, so what's interesting in this paper, they found that loss of function mutations in the cohort in the UK Biobank are associated with increase in body mass, but people with gain of function mutations are actually leaner, so they lose body mass. So it's, I don't know whether there will be obesity drugs for using this target, but this is where it's going, right? You have to know the direction of the effect. Okay, these are coding mutations. Is anybody tracking time when we started? I have the whole. Unless I'm being stopped, I keep going, okay? Like I keep going. If you're tired and you're in need of coffee or something, just let me know. Okay, so I'm moving to non-coding variation. Yes?
Can I ask, do I understand correctly when you told about this compensating mutation in fusional reinforced?
Yeah.
You said that they're not necessarily close in
sequence. In structure, it's a complicated business. We didn't have structure for those proteins, but at least if you look at the annotations, they are not all in the same domain. So I'm pretty confident, like in this story, I'm pretty confident that I wouldn't be able to sit and design, like within today's design method, to predict it. We did try to use structures to find compensatory changes. It doesn't work very well, so I think there is a lot we don't understand about these effects. They are probably transitive. So the easiest way to think about it, like, look, this is a receptor, right? That's classic GPCR. We know how it works. We know that conformation of this region is linked to conformation of this region, because this is how receptors transmit the signal. They are divided by the cell membrane. So predicting it from simple contact or electrostatic interactions or something like that, this was the original idea. So 10 years, I'm dating myself, 20 years ago, when we computed, they did this hemoglobin slide. So this hemoglobin slide is actually 20 years old. I believe personally that you can compute in a favorable energetic pairing and disfavorable energetic pairing in the structures, and I played with that quite a bit. Nothing got published because I never could get it work well enough. Don't know why. So it seems to be a harder problem than it looks on the surface. Again, maybe in my hands. Maybe there are smarter people who can do it much better. Yeah?
So these days, I see many sort of perspectives on DMS data. They say that, okay, we can use DMS data as the grand truth to get rid of all complications of the ClinVar and
Yeah. I don't believe that, by the way.
Yeah, this is what I was going to say, because in DMS you are only testing one aspect of function, right? However, in evolution is a process where there are many factors involved, selection process. I would like to know your comment on that.
Yeah, so it's an interesting question. So it's I have a colleague who went to the dark side and became an important person in the pharma, in the industry. So he calls me once and he said, okay, so we used to run the simulations for rare variant studies, and he's saying, okay, so shall we try to do DMS for every human protein, to run tests against every human phenotype in a large sequencing study and actually figure out every single drug target in the world? Are we powered enough to do this? Right? Running DMS for every human protein is a lot of money, so you have to think about this very seriously. I think there are a bunch of issues. So one, what you mentioned, you measure one aspect of function. You never know whether you're localized to the right location, because function is a lot of things, right? It's localization, it's stability, it's the proper binding. Sometimes you get some nonsense because you block antibody binding site and you get DMS data completely misleading, or any functional data completely misleading. So this is number one. Number two is noise. There's a lot of noise. And then, even if you get the direction of the effect, what's interesting, right? So computational methods is hard because with computational method I don't know the scale, right? Here I know the scale. I know this is a large effect, this is a small effect. But that's at the molecular level. How this scale is being transformed into phenotypic, right? So for example, this is MC4R. I think this is related to risk of diabetes and obesity, right? How is this effect translated into human BMI? Right? We don't know from DMS experiment. There is zero information. The last part, it's obviously context. It's not in the nature of context. So there are many issues. I would not trust DMS as standard of truth. I think current American College of Medical Genetics guidelines, you don't use it as standard of truth for pathogenical assessment. Classically, standard of truth come from human genetics, right? So if I see multiple de novo mutations of the same nucleotide in people with the same phenotype, I know this is real. If I have a variant segregating in multiple pedigrees through multiple informative meiosis, I know this is real. I know human genetics tell me this is real. If I see experimental evidence, it has to be contrasted with the human genetic evidence. At least this is my take on that. Again, can you, sorry, long answer, but one last comment. There are computational groups now suggesting that with some DMS data and some computational predictions, you can actually move the ROC to clinically actionable guidelines. So you can be, for a tail, for a subset of very strong predictions, you can be as accurate as current clinical guidelines. And they try to identify the subset. And if numbers will work out, I think I'll probably support that. Clean bar is hard because, like, you realize what's happening, right? So you have, for example, new mutation in the BRCA gene, and a patient would undergo very traumatizing surgery, right? You don't want this decision to be based on an experiment with a lot of noise or on a computational prediction. Like some of these decisions are dead serious. Some are not. Okay, shall we still move to non-coding variants?
Yes.
Okay. Non-coding variants. Well, you probably know the gist of the story. The human genome got sequenced. There are very few genes, a lot of non-coding space. Then you're looking at conservation between species. You look at where common disease heritability resides. It's all in non-coding variants, and potentially it resides in regulatory sequence. Again, I told you I'm not talking about splicing in this talk. I know splicing is important. Every time you're saying everything in regulatory, people are asking what about splicing. I know splicing is important. I'm not talking about it. You have promoter sequences, you have distal enhancer sequencers, you have insulators, you have silencers, you have a whole bunch of different things, right? So this is all regulatory machinery, and this is probably where the action is. How do we know this works for human genetics? We have very few examples where we know what's happening, right? So one is this gene OCA2. It's an eye color gene. Eye color is not truly Mendelian phenotype, but if you're a homozygote for OCA2 loss of this enhancer function, 98% chance you have light eyes, and green or blue on the graph. Right? So it's a very large effect. This is one of the examples of human alleles that are non-disease and large effect. And they're very few, by the way. In the human population, there are very few, because of natural selection and other forces we discussed, there are very few variants that are a large effect on non-disease, and one of them happens to be this one, and we know that this is exact regulatory mutation, right, a very large effect. Another example of human non-disease very large effect is lactase, lactase gene, right, the story goes that before dairy farming, grown-up humans could not process lactase. And then the regulatory mutation became very beneficial because it allows humans to consume milk products with no problem, and still human population segregates this variant. And this variant turns out to be non-coding, and what's important, it's not even within the lactase gene, it's in the intron of the neighboring gene. That's a distal enhancer variant. It's a large effect, we're absolutely sure about the mechanism. So we know these things happen, but we know very little outside of this. Now, what we know, and I mentioned this, we now have a whole bunch of mammals in the Xenome Consortium, and on top of this we have additional primate genomes in the paper I just mentioned. This is 2023, the other paper 2023, all of those genomes just showed up. Like, we're literally starting analyzing those. And if you do heritability partitioning, now what is heritability partitioning? Like, very quickly. We discussed that we believe in this normal distribution world of statistical genetics. Everything is about variance. Variance is decomposable. I can ask, I don't know where functional, because of LD, right? My problem is confusion. I don't know where my biologically significant variants responsible for high blood pressure, heart disease, where they are in the genome. So for any nucleotide, I don't know whether this is functional or not, right? I can run whatever association studies, but because everything is correlated to everybody, right, I have no idea. But I can run a sophisticated model. I can say, or in this case, like this people can, or Stephen Gazal can, and other people I would show example of work. I can ask how much genetic variance is due to SNPs under this annotation. Turns out that if I look at the distance, conservation with especially primates, what's interesting, especially within primates, explains a lot of signal for a lot of human disease, right? So these are positions that are conserved or within conserved segments of the sequence with every primate species. And this is what explains most of heritability. That's the strongest signal we have. Attests to the power of evolution. Evolutionary genetics is great. The problem is I have no way to interpret it. Right? I know it's conserved among primates. What in the world does this science do? I don't know from this. So the next observation is more directly functional. And this is the observation that a lot of these functional variants fall into regions of open chromatin. This is based on DNAhypersensitivity assay. And those are regulatory sequences. Now, I'm a little bit confused about this because I know that every single regulatory sequence is open chromatin. A lot of people believe that every open chromatin mark is a regulatory site. I don't know whether we know this, but there are some people who believe that. And there are chromatin modifications, right? So a lot of this epigenetic marks that mark regulatory elements. A lot of heritability sits there. Now, this is the story how I was blocking progress of science. Back in 2012, my collaborator John Stevens calls me up and says, you know what, I'm looking at this GWAS peaks against our experimental DNA signals, and I'm finding that they line up. So our DNA signals explain GWAS signals. This is like 10 years ago. And I'm like, John, everybody can eyeball anything, right? So GWAS signals are correlated, DNA signals are correlated, everything is lining up with everything, and it's not really the case. So we figured this out, so this is the first author was Matt Morata. Now I would show this is the most telling thing. How many people know what QRS duration is? Yeah, I see some people with medical background nodding, but not a lot. So, so QRS duration is when there is EKG, right? So we're measuring electric function of heart. Doctors are not very good with infinite dimensional objects, right? So the curve is infinite dimensional objects. So they give the curve the letters P, Q, R, S, whatever. And there isn't one duration. So this is specifically heart phenotype. There's genome-wide association study. You look at positions at p-values, enrichment of significant variance in regulative regions, and you find them in heart, specifically in heart. And that's, I think, is a very strong evidence that the signal is real. Right? You're actually guessing the right tissue. The effects are very small, there are very many variants, but they are all in regulatory regions, specifically in heart. If I look at multiple sclerosis, I get immune cells. If I look at Crohn's disease, I get immune cells, and this is what I expect. Later work by Alkas Price and Sasha Gusev, so this is heritability partitioning. They were pretty generous, but they are saying that 80% of all variation in underlying human disease sits in open chromatin. So that's a regulatory signal. This is more recently, again, heritability partitioning and conservation explains a lot. Coding, of course, is very enriching, but it's a small amount of real estate. Still, even in the joint model, chromatin and regulatory related features are quite rich. There is application of all of this, right? People try to use this enrichments in fine mapping, trying to find what variants are truly biologically functional in this chromatin block. Okay, I have the last segment. Am I still okay on time or not? I'm fine. Okay, good. So, now there is this new idea that we can use molecular phenotypes. And long-standing studies in the field were EQTLs. How many people know about EQTLs? Not a lot. I didn't include specific, okay, some people know a little bit. I didn't include specific slides, and then I'll try to hand wave about EQTLs, right? When I'm running association study, I can run any phenotype. I can run heart disease, I can run diabetes, I can run transcription factor binding, I can run gene expression. So the idea is that if my variants are regulatory, that the variant that acts through changes of expression of a certain gene, right, this genetic association should line up with genetic association where my phenotype is expression of the gene. So EQTL are expression QTLs. They are QTLs relevant to expression. Now this is the whole logic of the field, and then I'll talk about our recent paper where we believe everything is much more complicated than people think. So the idea is this. I have a GWAS peak. So the original idea was that I have to find out what's happening. I should move it to controlled model system, fish, mouse, yes, whatever, cell culture. And then move to biochemistry and figure out what was the biology. Right? That's a very difficult proposition because imagine you have a region of 150 kb where you change height by 0.1 millimeter, and you're saying, let's do experiments on this. Like, what kind of experiments are you going to do? Or, so then there was this idea that I used to call human genetics all the way. And the idea is that you measure phenotypes at the molecular level. So I have GWAS peak, and then I measure some of the phenotypes. And for example, I measure something at the cell level. And then I measure gene expression as another phenotype. And then I measure, for example, methylation or transcription factor binding site. So if I see that my association signal for molecular QTL coincides with my association signal for expression QTL with endophenotype and then with GWAS peak, this is much more tractable. I can bring this to experimental lab because I know which gene expression is controlled, how it's controlled, how it's presented in the cell system, right? So this is something I can work with. Now the problem is that I think that my genotype causes expression or endophenotype and causes phenotype. But because of LD, I can have two different signals. One causes endophenotype, one causes phenotype, and they are in correlation. So I have to figure out what to do. So this poses the problem which is called colocalization. So this is what's happening. This is some computer simulation, some tune did in the lab a long time ago, related to a specific method. That's not very important. What's important is that I'm looking at one trait. For example, it's a disease trait, right? And all of these guys are significantly associated. I don't know which one. In this simulation, this is a true positive variant. I don't know that this is a true positive variant. And this is another phenotype, for example, gene expression. This is the association. And again, in this case, for example, this is correct positive variant. Now I don't know about it. But what I can do, I can develop statistical machinery to test by looking at the joint distribution of these effect sizes and linkage equilibrium. I can test whether there is the same positive variant that drives both of this or not. Okay, so I can colocalize the peaks. And there are different methods called caviar, we developed JLib at some point, again, Sanchon in the lab, when he was in the lab, developed it. Colocus, I think, is the most popular method right now. Okay, so we try to test how this works because this is the hobby horse. And the way we did it, we looked at complex trait variants in genes that have large Mendelian component. So I have to know which genes are truly causing disease, right? And for example, I have monogenic version of diabetes, and I have a GWAS peak for diabetes. I have a monogenic version of inflammatory bowel disease for a specific gene. I have polygenic peak there. We redid all genome-wide association studies, removing coding variation, so we know that mostly Mendelian variation is coding, and we will look at regulatory. Again, the model is that I come to the right gene using coding variation, and I'm trying to test the non-coding variation expression. So the hypothesis is that there will be changes in gene expression for genes which I know are involved in the trait and have peaks around them in the non-coding region. So these genes include, for example, I discussed a lot LDL receptor. So LDL receptor, if I exclude the coding region, has massive non-coding peak in association with LDL. If I run genetic association with LDL, I find LDL receptor under the peak. I bet my money that this is the gene, right? So the LDL, there are multiple genes maybe under the peak, but I believe LDL receptor drive the peaks. I see, for example, breast cancer association study. Estrogen receptor is under one of the peaks. I believe estrogen receptor drives association with breast cancer, right? So that's the logic. So we ran colocalization methods, 3 of them, transcriptome-wide association studies, TWAS, it's called fusion, and analysis of chromatin marks. Chromatin marks work to some degree, it's just hard to find the right gene. And we just look at the closest gene to the peak, right? So again, colocalization between GWAS and gene expression, right? So there are multiple genes in the locus. We're saying this is correct because its shapes of the peak is the same. It's much more complicated than shape of the peak, but I'm kind of glossing this over. And you can run just genetic correlation with the locus. It's called transcription-wide association study between the gene and the expression. This doesn't really correct for LD, but this corrects for the possibility of multiple variants in the locus, and generally has high power. So the issue is, what's happening is very bizarre. So I have the genes that I believe are included in disease. These are found by rare variant burden testing. These are Mendelian components. Most of them have non-coding associations. Very few of this have any evidence of changes of expression of the right gene. I really don't know why. I mean, this is very bizarre because we know the genes are regulatory and they don't colocalize with expressions of the correct genes. Chromatin is doing actually quite a bit better, but it's very hard to find the right gene using chromatin because I just know that it was chromatin association peak. This is the full table, right? So these are 3 colocalization methods. So mostly, these are the classes of the genes. This is whether it's a QTL or not. There is a QTL in the region. And then whether this colocalizes, and the last column is chromatin, and most of them don't. Okay, so why? Is it because we're not looking at the right cell type? Maybe we're not looking at the right context. We brought to the gene through coding variant. It's not context specific, but maybe expression regulated is interesting. Maybe it's about certain developmental stage, maybe certain environmental condition. Or maybe there is non-linear relationship. So what we're measuring total amount of the message, and if but the effect is on the rate of transcription, right? So if there is non-linearity built in, maybe this propagates. So, the other thing, we call them red herring QTL. Sorry, it's an American expression, I don't know if people know it. And so we do have a QTL. So what's surprising is I do have a variant that changes expression on the right gene. It just doesn't change the phenotype. Another variant does. So what we're doing right now, and yes, there are multiple papers also questioning the QTL paradigm here. We have to get out of this. So one way to get out of this, I promised one of you 2 single cell slides at the end of my, like, these are 2 last slides, but they are single cell because I specifically promised to include them. So this is the paper by Nathan et al. From Shamil lab. And what they look at, they are finding these interactions between positions. So this is single cell UMAP. And you're saying, oh, there are SNPs that are EQTLs interacting with position of the cell in the transcription map. Right? So here there is the effect, and here there is no effect. So it may be that specific groups of cells is responsible for the expression change. So Jonathan Mitchell in my lab, he developed a new method looking at association by going through, this is specific for autoimmune diseases, this is rheumatoid arthritis association, and this is the GWAS and the locus, and you see exactly the same peak, but only in this group of cells. Now there are many questions, I'm not sure these groups of cells make sense biologically, but we find the statistical subsets of cells that show that genetic control of expression correlates with the effect of the phenotype. Still not a lot of them, we're finding more. And I'm not sure that what we're finding is correct. This is, again, this is very early work. But this is where we're moving, so the whole idea that the simple analysis of regulatory function didn't work in our hands. And again, the QTLs are great, right? I don't see any other, I don't have any other biological hypothesis. But we probably have to chase a context specificity, and we probably should move to this multidimensional spaces of single cell data. Guys, we're amazing today's. This is my last slide. I'm done.
So we have finalized this excellent series of specialized lectures with Professor Shamil Sunyaev, who indeed brought to this auditorium the spirit of Harvard. And thank him very much for all he has done for this. And thank you for all you for participation and coming and joining us. So I hope you will.
Thank you very much.